Good morning to one and all, respected sir and all my dear fellow mates. Today I am Maiolica Bandyopadhyay from section IMT 6C of roll number 69, going to give a talk about a very interesting biotechnological methods working and its uses. And the title of my topic is CRISPR-Cas9, a tool for genome editing to study human genetic diseases, which is extracted from this review paper whose title is shown in a snap form just below my topic. And my presentation goes in this way. First, this particular question always comes in mind before talking about it in detail. What is CRISPR? CRISPR stands for clustered regularly interspaced short palindromic repeats, and it is the family of DNA sequences which are found in the genomes of prokaryotic organisms such as bacteria and archaea. Here in the figure given on the right hand side, we can see here in the lower strand CTAG sequence whose complementary is given here as GATC. And while doing the mirror image of GATC, we can see the same as CTAG. Hence, the palindrome comes, and these sequences are repeated just as in this in the right hand side, which is why the name repeat came. The word repeat comes, and hence the name comes as CRISPR. And Cas9 or CRISPR-associated protein is an enzyme that uses CRISPR sequences as a guide to recognize and cleave specific strands of DNA that are complementary to the CRISPR sequences. Other enzymes with similar cleaving functions are Cas12a and Cas13. And here the discussion will be more with CRISPR with Cas9 enzyme together, with which they form the basis of CRISPR-Cas9 technology. Genome editing re-emerged in 2012 with the CRISPR-Cas9 technology development, which is a genetic manipulation tool derived from the defense system of certain bacteria against viruses and plasmids. And this method is versatile in a wide variety of experimental models, including cell lines, laboratory animals, plants, and even in human clinical trials. It is a powerful tool for biomedical research and provides hope for curing and correcting some inherited and genetic diseases. And the process that allows a permanent modification of the genomic target sequence can repair the DNA damage caused to DNA. In the year of 2020, during the pandemic, the renowned scientists Jennifer Doudna and Emmanuel Schumpeter, whose images are shown just below, they were given Nobel Prize in Chemistry for the discovery of CRISPR-Cas9 genome editing technique. And I have also read this book, The Code Breaker, written by Walter Isaacson, where we can find the life history of Jennifer Doudna and how she struggled to know the code of life and how the working based on CRISPR-Cas9 they did. And I highly recommend this book to read and know about the modern biotechnology concepts and their uses in an interesting way. Now let's discuss the mechanism of CRISPR-Cas9. CRISPR was discovered in Escherichia coli in 1987 as a series of repeated fragments of 29 nucleotides in length with variable sequence fragments of 32 nucleotides. Interest in the CRISPR system and its associated Cas9 led to the discovery of similar short repeated palindromic sequences of 24 to 40 nucleotides in several groups in prokaryotes. The CRISPR-Cas9 system consists of directing the Cas9 nuclease to create a site-directed double-stranded DNA break using a small molecule as a guide. So here we can see inside a cell a complex forms by Cas9 protein with guide RNA, which then attaches to a matching genomic DNA sequence adjacent to the spacer, which is the yellow segment shown here. And then the Cas9 RNA complex acts as a scissor and cuts the double strand of the DNA. And at last, the programmed DNA would be replaced at the cut portion. Now let's talk about genome editing working using CRISPR-Cas9 system tool. The complex formed by Cas9 and guide RNA, as shown in the figure, just close to the protospacer adjacent motif site, by which double strand break is generated in the target site, and that could be repaired by two methods shown here. One is non-homologous end joining and other is homology directed repair. And non-homologous end joining is prone to error as it results in insertions or deletions or frame shift that causes gene knockout by interruption. And homology direct repair is error free as here a donor DNA with homology in the ends is provided, and this DNA can be inserted to the target site to modify the gene, and gene correction occurs by introducing the nucleotides and leading to frame shifts or insertions of complementary DNA. Here are some diseases which can be cured and corrected using genome editing by CRISPR-Cas9 system. And due to the time constraint, I will discuss only the first 5 diseases. And here in the right hand side we can see the CRISPR image. CRISPR-Cas9 in pulmonary and gastrointestinal diseases. Cystic fibrosis in intestine is caused by the homozygous delta 508 mutation in the CFTR gene, which causes the deletion of phenylalanine at residue 508. And using CRISPR-Cas9 in vitro in intestinal stem cells of cystic fibrosis patients, it can be cured. While the CRISPR-Cas9 will create the double-strand break in the targeted site as shown here, just here. By targeting vector, the mutation will be corrected in that 508 site. CRISPR-Cas9 also shows potential increase requiring a liver transplant like in drug therapy refractory metabolic liver disorders. The main cause of hematologic disease is the mutation in the beta globin gene, and the targets for curing this disease is to achieve homologous recombination in beta globin gene in induced pluripotent stem cells through CRISPR-Cas9 and donor sequences, and also to insert a mutation for generating the benign genetic condition in which the mutations attenuate a shift of gamma globin to beta globin, resulting in high level of fetal hemoglobin expression, and which results in the relief in clinical signs of hematologic diseases like beta thalassemia or sickle cell diseases. And also making mutations in BCL11A erythrocyte enhancer will be another method to increase fetal hemoglobin expression. In similar way, the cause and solution of sickle cell disease and beta thalassemia are mentioned here specifically, where the mutation of sickle cell disease was glutamine 6 valine mutation, and the mutation for beta thalassemia are some point mutations where expression or suppression or deletion type of point mutation had taken place. We all know that Anopheles mosquito is the main vector for spreading malaria, and CRISPR-Cas9 will be used to provide a deleterious trait of the vector, where here the CRISPR-Cas9 system will be used to confer a recessive female sterility phenotype upon disruption of 3 target genes, which I also showed in the diagram on right hand side. This study demonstrates that disrupting AGA P007280, one among the 3 targeted genes, alone was sufficient for a successful gene drive targeting female reproduction in an insect population. And here, primary immunodeficiency is a group of heterogeneous, rare chronic disease whose causes are the inappropriate immune system function or non-functioning effect. It is characterized by the absence of mature T, B and natural killer cells. And CRISPR-Cas9 has been used to generate knockout models that had not been susceptible to efficient genetic modifications for study of immune system diseases. For investigating allergic diseases, CRISPR-Cas9 technology has been used to examine certain genes causing allergy, like MUC18 gene, also known as melanoma cell adhesion molecule. And through this technology, the knockout of MUC18 gene was conducted in human nasal airway epithelial cell in the following manner, just shown in the figure on right hand side, which is the first line of defense against environmental factors like pathogens and pollutions. And this is how that allergy will be reduced or cured. So I conclude CRISPR-Cas9 technology is a very powerful tool for genome editing to fight against many genetic diseases. And here are some references. And I would like to give a sincere gratitude to Dr. Snehashish Mishra sir for encouraging us to do research study on topics of our interest and make use of seminar day as the day of making all other future biotechnologists understand and know the topic with the concepts I have obtained. And I would also like to give a sincere gratitude to my parents who encouraged me to go ahead for the topic I selected for today's presentation, and I hope the concept I gained would be useful in my further future works as well as for others. Thank you for your patience listening.